hopefully. Yep, we're recording. Let's go back to the PowerPoint. And so how is an action potential generated? When we look at an action potential, the first thing that opens as input from dendrites causes a voltage change by sodium, the sodium channel opening. Sodium rushes in, and that causes the spike. And this is a measurement of the change in voltage as a nerve membrane allows ion sodium to move in and then potassium to move out. So as positive charge moves in with sodium coming in, depolarization means a change from a negative inside cell voltage to a positive cell voltage, and that change is called depolarization. The sodium channels close, and then the potassium channels allow the high concentration of potassium to drive potassium outside the cell. As the positive charge leaves, that allows the inside of the cell to repolarize and to hyperpolarize, and it determines how quickly a nerve can respond to the next signal. So this action potential is a result of sodium moving in, potassium moving out, and it perpetuates itself, or it moves down the axon away from the cell body. Okay, let's keep going. So there's two types of propagation: unmyelinated axons. The nerve signal moves along the surface of the nerves. If you have myelination where the nerves are insulated with fat, then the nerve signal jumps from along the nerve, and it's very, very much involved in motor neurons, and the nerve impulse actually jumps from one fat covering to a gap in between the next fat coverings. So action potentials, notice when sodium comes in, the inside of the nerve in front of it is all positive. So as that sodium comes in, that sodium is going to move toward these negative cells, and that's going to allow the sodium channels to close, and then the potassium channels to open, and that's how the impulse moves away from the cell body, and the nerve impulse travels along the whole surface of the nerve membrane. If you have saltatory conduction, then the sodium will come in, and then it cannot go in this internode. So the signal basically jumps from this gap here called the node of Ranvier, and it jumps very quickly. And these motor neurons that have myelin are insulated, and the conduction speed is greatly enhanced. So you can see the local current is only going to be pumped back by the sodium. I got to turn this off. All these people are calling me. Okay, so myelinated, and these are myelinated. They are insulated by fat. The nerve impulse jumps. The nerve spike jumps from node of Ranvier to node of Ranvier. And so that allows these positive charges to be attracted by the negative charge, so that there's a long current flow that, because of this insulation, can jump. Saltatory conduction means to jump. So the action potential or nerve signal jumps from these gaps where there's no fatty insulation. Okay, we'll keep going. So action potentials are electrical signals that are known as action potentials. The most important information, vision, balance, movement is carried by these myelinated axons. So what's a synapse? A synapse is the gap between the presynaptic nerve ending and the postsynaptic membrane. So all skeletal muscle has to be stimulated by motor nerves. So that's why it's called the voluntary muscular system. All communication between nerves is going to send an electrical signal down the axon. Calcium is going to move in. That's going to cause these vesicles to fuse with the postsynaptic membrane, allowing the sodium channels to be open, and that leads to depolarization or positive voltage change from the presynaptic neuron to the postsynaptic receiving location based upon nerve. It could be a nerve, it could be a muscle cell. So the synapse allows for a neurochemical to be released, and everybody should rejoice because without neurochemicals causing communication between the presynaptic neuron, because the gap here in the synapse, the communication is carried by a chemical, the acetylcholine in these synaptic vessels, and those chemicals open up channels so that the movement of ions inside the cell can have an electrical signal move along the nerve. So thank God for synapses and for neurotransmitters, because we couldn't get high. Cocaine wouldn't work. Marijuana wouldn't work. Speed wouldn't work, because all of these drugs, a lot of drugs, cause changes in how much neurotransmitter is released and affects how nerve signals are carried across synapses. So when there's an electrical event causing the nerve signal to come down the axon, that signal with potassium, with calcium coming into the nerve, causes neurochemicals to be released, such as acetylcholine, dopamine, norepinephrine, epinephrine, various, those are the primary neurotransmitters. And the modification of those levels of neurotransmitters and how much is released, that's what getting high is all about. Okay, let's stop there. Go back to here. Stop recording. Stop recording.